Good afternoon. Today we're going to think about why calico cats, tortoiseshell cats, are all female. And to do that, we need to think a little bit about the genetics of sex. Now, the existence of sex itself is an evolutionary problem. We're not entirely sure why sex exists. There are various theories, and most of them centre around the idea that meiosis, through recombination, meiosis and sex together allow you to mix genes with those of other organisms, so that the offspring have a mixture of genes from two different parents rather than from just one parent. Exactly why this has been selected for by natural selection, we're not quite sure. There are a number of theories. Two of the leading ones are something called the Red Queen theory, hypothesis, and something else called Muller's ratchet. Those aren't directly relevant to the module. But when you look at the evolutionary theory behind sex, it predicts that you will have two parents, which is not immediately obvious. Why not have three or four? If there's an advantage to mixing genes, why not mix genes from as many parents as possible? Why two? And it predicts that two parents, and there are two possible strategies that these parents can adopt. One is to make gametes, that is the thing that you put your genes into to be mixed with the genes of the other organism, the other parent. One strategy is to make lots of gametes, but not to invest much effort in making sure those gametes survive, and that's the male strategy. The other strategy is to make fewer gametes, but to invest much more effort in making sure that those gametes survive into adults. And that is the female strategy. Now, so that means that there needs to be, when the evolutionary theory is considered, it predicts that the best bet for an organism is to pick a strategy right at the start and stick with it. And in plain language, that means that organisms start male and stay male, or they start female and stay female all the way through their lives. There are some exceptions to this. There are always exceptions to everything in biology. So there are organisms like the slipper limpet, the Crepidula fornicata, which changes sex throughout its lifetime. But by and large, most organisms stay the same sex. And this raises the question of how then is sex determined? Way back in the mists of time, sex was probably determined in all organisms by environmental cues, and it still is today in some animals. And most famous example is crocodiles, where the temperature of the egg during incubation determines the sex of the resulting offspring. So if the temperature is between, I think, 31 and 34 degrees, the offspring will be male. If it's outside that range, it will be female. However, the second way of instructing an organism to develop as one sex is through what we call genotypic sex determination, which basically means there's a gene for sex. And this happens now in probably the majority of animals through the evolution of what are called sex chromosomes. And sex chromosomes have evolved independently in different organisms, different eukaryotic lineages, independently in different animal lineages as well. So insects use the X0 system of sex determination. Mammals use the XY system of sex determination, and this is the chromosomes that were discovered through karyotyping by Wilson and Stevens, who are on your screen now, the X and Y chromosomes. And and reptiles and birds use the ZW system. It's effectively the same as the XY, only it's the other way around. So the homozygote is actually the male and the heterozygote is the female. So genotypic sex determination requires the evolution of a region of the chromosome that specifies what sex the organism will become. Now, there are two things that are relevant for this lecture. The first is that sex chromosomes have evolved from autosomes. That means that they evolved from chromosomes that had lots of other genes on as well. One of the genes on that autosome became the sex determining gene. So there are lots of other genes on the autosomes, or sorry, on the sex chromosomes. This leads to the phenomenon of sex-linked diseases. So many of those other genes, and on the human X chromosome, we're talking about 1,000 genes in total. The majority of those genes will code for things that are not sex determining. And so you have the example that we had in the lecture was that of haemophilia. You can see on your screen now the Russian royal family at the turn of the last century, and the little boy on the bottom right was a haemophiliac. The haemophilia is a sex-linked disease because the gene that is responsible for haemophilia is on the X chromosome. It's a blood clotting factor. If you have a mutation in that gene and you're female, then most of the time on your other X chromosome you'll have a working copy of the gene, so you don't have any disease phenotype. If you're male, on the other hand, you don't have a balancing copy of that gene. So you've only got one allele for that particular gene. If that allele happens to be not working, as is the case in haemophilia, then you'll develop the disease. So certain conditions are sex-linked. And in the case of the Russian royal family, that is thought to have been because Queen Victoria had a mutation in the egg that went on to become her. So there is no balancing working allele on the Y chromosome, and that leads us on to the second important thing about sex chromosomes, which is that the important thing about sex chromosomes is that they code for a distinction between two organisms rather than for a particular protein. They code for maleness or femaleness. And because you want to maintain two sexes distinct from each other, there is no advantage to mixing the male and female genes. There's no advantage to having something that is halfway male, halfway female. So that means that sex chromosomes, the X and the Y chromosome, have evolved suppression of recombination. So the recombination that occurs between all the autosomes at meiosis that shuffles genes about, moves genes from one homologous chromosome to the other, that is suppressed between sex chromosomes. Now, think back to the when we were talking about meiosis and recombination, and that plasticine video that I put on another video. When chromosomes pair up and recombine in meiosis, you get a holiday junction formed, and that holiday junction can resolve in one of two ways. Either you can get crossover product, which is what causes the transfer of material from one homologous chromosome to the other, or you can get non-crossover product, in which the two crossed over sections of chromosome go back to the chromosome they started on. I said that I said in that lecture that that non-crossover product that resulted from chiasmata formation during meiosis was a mechanism for DNA repair. Now I won't go into that too much, but what that means is that it means effectively that when homologous chromosomes pair up during meiosis, that can be used as a way for effectively one chromosome to repair another. So if, now assuming two X chromosomes, if one X chromosome has a mutation or has been damaged, it can be repaired effectively using information from the other. Because recombination has been suppressed between the X and the Y, that means that the Y chromosome, if it picks up mutations, the Y chromosome can't be repaired. And that means that the Y chromosome accumulates mutations and genes on the Y chromosome stop working. We say that the Y chromosome becomes degenerate. And in fact, if you look at the Y chromosome, most of the genes on the Y chromosome no longer work. Now, think back to another lecture, the lecture on aneuploidy. We saw that in general, the amount of gene product in a cell is proportional to the amount of DNA that codes for the gene product. This immediately raises the problem of what happens with X chromosome gene product. Now think, in females you've got two X chromosomes. Each of them has got a number of genes that are not sex determining or sex coding on them, and they're all being expressed. In males you have one X chromosome and one Y chromosome, and all of the genes on the Y chromosome, most of the genes on the Y chromosome, have now been switched off, they've degenerated. So potentially you have twice as much gene product coming off the X chromosomes in females as in males. We know from the aneuploidy lecture that mammalian development will not support this. So how do we get around this gene dosage problem? There are three ways. One, we could run both the X chromosomes in females at half power. And this actually happens in some organisms. It happens in the nematode worm, Caenorhabditis elegans. Mechanism number two, we could actually run the X chromosome in the males at twice the speed. We could hyperactivate the X chromosome. And we see this in Drosophila, the fruit fly. So if you look on your screens now, you'll see on the left hand side you'll see the female chromosomes in Drosophila, and the level of green indicates how much gene activation there is. And you can see that all of the chromosomes, there's 4 chromosomes there jumbled together, they're all running at about the same speed. If you look on the right hand side though, you can see that these are now the chromosomes in male Drosophila. You can see that one chromosome is running faster than the others, and that is the X chromosome in Drosophila that has been in the male Drosophila that has been hyperactivated, so that it is generating as much gene product on its own as the 2 X chromosomes are in the females. The third mechanism is that we could switch off one of the X chromosomes in the females, so that both females and males only had one active X chromosome. And that's what happens in mammals. The X chromosome is, or one X chromosome at random in females, or in every female cell, is converted into heterochromatin. It's coiled up, it's wrapped up, it's condensed too tightly for the machinery of transcription to get to it. This process was first proposed by a lady called Mary Lyon, and the process is often called lionization after her. X chromosome inactivation, or lionization. The condensed X chromosome, the inactivated X chromosome, becomes something called a bar body, and you can see this down a microscope. It's a very dense body. Now, in mammals, this X inactivation happens about 12 days after conception, when the cell is about, the embryo is about 100-200 cells. Because each individual cell in that 100 cell stage embryo, each individual cell will give rise to cells in the adult organism that are next to each other. And that means that in that 100 cell embryo, 12-day-old embryo, there is random inactivation in all the various cells, which means that the 100 cell 12-day-old embryo becomes a mosaic. Some of the cells have one X chromosome inactivated, other cells have the other X chromosome inactivated. When that embryo grows up to the adult organism will reflect the mosaicism of the 12-day-old embryo, which means that adult mammalian females are mosaics. You can see this here in a picture of a adult human female. There's a condition called, I think it's called sweat gland deficiency, and you can see that the skin on this girl is patchy. One patch of cells has derived from a cell in the embryo that had the mutant sweat gland gene chromosome inactivated. Now that brings us back to the tortoiseshell cats. In cats, coat colour is determined by a gene that is on the X chromosome, and that gene can have a couple of different alleles. One allele gives rise to an orange colour, one allele gives rise to a darker colour. If the X chromosome with the orange colour is inactivated, the cell will be dark. If this is in a heterozygous cat, if the X chromosome with the dark allele is inactivated, then the cell will be a lighter orange colour. So the patches on the tortoiseshell cat simply reflect the pattern of X inactivation that happens in the embryo. There is a further complication, which is that obviously white patches on the tortoiseshell, that's another gene that is also involved, but we won't really go into that.